you have your whole complex of your CRISPR-Cas9, and then it's gonna make this cut. So I don't know if you can see it super well, but I don't know. I don't even think. Oh, I can. I don't know if that helps anyway either. Okay. Anyway, I'm just gonna move along. Y'all will get these online. So you have your DNA, and so your CRISPR-Cas9 complex will just come in, and it's gonna just start to separate your double helix into your two separate strands. And then from there, it's gonna make that cut where it sees the PAM. So I don't think you guys can see that. It's like these orange little nucleotides. And that's where it's gonna do it a few upstream, and so it's gonna make that cut a few nucleotides upstream. And so then it does that, it snips it, cuts it in half. There's the PAM you see there, or the flags, tags, whatever, airplane directors, whatever you want to call it. And so then it splits off, and then that's where you can either delete a gene, or you can add a gene or alter it in any way you kind of want to. So, and that it just kind of shows down here. I don't know if you guys can see that. There it is, I think. But anyway, that's just kind of where you can like insert it right here. And that's like showing the gene with a new sequence, or just like completely disrupting that gene. So that's just kind of like the basis of like how it works in general. It's a pretty simple technique. But anyway, so my next thing is like, how does this kind of work within the realm of my research that I looked at? And so there's a thing like for my research, I'm going to go ahead one. So like my research that I looked at is talking about lentiviral CRISPR-Cas9 vector and how it's like mediated the MIR21 gene editing and like kind of how that inhibits, like basically just kind of how it's inhibiting the epithelial to mesenchymal transition in ovarian cancer cells. So basically, like, is there an interaction with like the MIR21 or like kind of like the microRNAs? So is there like any sort of impact with that? So kind of going back, I hope you guys can see this. I really can't. I don't know. Anyway, sorry if you can't. Anyway, I lost my train of thought. Anyway, okay. Excuse me. Anyway, so the lentivirus, to kind of like get some definitions down, the lentivirus is a family of viruses that are responsible for different notable diseases. So like cancer and HIV, which I keep saying because cancer is like kind of like what I did for this thing. And so that's just kind of like what that incorporates. And so lentiviral CRISPR-Cas9 basically just means like it's its way of inserting these diseases into, or like these disease mutations into the different parts of the DNA to look at for further research. I don't know why that was so hard for me to get out. So microRNAs, which I had like just said, it's also known as miRNAs. Yeah, miRNAs, or like my specific one that I was looking at in this study is MIR21. So it's just a specific kind of microRNA. But anyway, microRNAs are a class of non-coding small RNAs that negatively regulate gene expression at the post-transcriptional level. And so miRNAs are transcribed and processed into precursor miRNAs, or like pre-miRNAs, by means of like a microprocessor, and it's called DGCR8, which I'm definitely not going to concern myself with because I just don't really like. It's just too deep of a level for me to get at right now. But anyway, so like, excuse me. So the MIR21 is a microRNA, like I said, and it's just like one of the most consistently unregulated microRNAs in cancer. And it's known to be like also one of the ones that are most known for being or like contributing to tumor metastasis. So that's kind of why like they targeted this specific microRNA, and kind of like why I'm also talking about it. So there's a quote within the research that said, CRISPR-Cas9 is a novel approach for loss of function studies by disrupting miRNA gene by disrupting the miRNA gene through introducing mutation in pre-miRNA sequences and disrupting miRNA expression. So that's just kind of like to summarize what I'm like saying. So like for like the precursor, like leading up to all this, I think that's all. Is that all of this slide? I don't know. This is really hard to see like backwards, and this backwards on my camera, and this is difficult. This is complicated. But basically, oh yeah. So basically, this like testing, is like used, or like they're testing this to see if lentiviral CRISPR-Cas9 vector efficiently or like effectively disrupts miRNA function. And so that's just kind of like what they're looking at in this study. Excuse me. So just to like give a little bit more background into like this whole thing. Epithelial to mesenchymal transition is associated to tumor metastasis and chemoresistance, which is kind of like, wow, like that's what, there's like 3 subsections of this, or 3 like parts of this, and that's kind of like what they're talking about in here, but I'm just focusing on that, like that one specific microRNA. But anyway, so epithelial to mesenchymal transition is associated with tumor metastasis and chemoresistance. Furthermore, the role of EMT in tumor metastasis and chemoresistance has been recently recognized, and is like kind of been a contributing factor to poor patient survival in ovarian cancer, which is actually what my grandma passed away from. So that's another reason why I was intrigued by this, is just kind of like what factors go into this, what factors can like be removed from this to increase that, because like most times it like ovarian cancer doesn't get diagnosed until it's like super too late, and so it's kind of like what can be done to like fix this. So anyway, they're basically just performing this experiment to see if CRISPR-Cas9 can disrupt that microRNA function and inhibit cells, like inhibit the cancer cell growth. Sorry, it's really cold in here, guys, so my nose is kind of running, but anyway, I'm out in my shed because it's the only quiet place in here. Okay, anyway, moving along. So this is kind of showing what the research was saying. Guys, I'm struggling. Uh-oh. Go back. So, oh my word. I give up. Y'all can look online. Anyway, I'm sorry. Anyway, so the figure 1, A and B, is just kind of showing like the mature micro R21 up top, and then the 4 micro R with the guide RNA sequences, indicated in the pre-MIR21 hairpin structure. So that's like kind of showing what these are, which I know you guys can't really see that. I can't work my computer very well backwards, but anyway, these are just kind of showing the differences of each of those like each of those 4 microRNAs. And so in C is kind of like what we're going to really focus on, C and D. And C and D, you have your control right here, your DNA ladder, which we just did this in lab 1 week ago, 2 labs ago. So I mean, this should be familiar to everyone. But this is a control, and this is your ladder, and here's showing your guide RNAs, your 4 different kinds of the microRNAs. And each of these, you can see that there's a mutation. And then if you look on the slides online, you can see that there is a mutation in guide RNA 1 to 3, 1 to 2, and 1 to 1. So like the sequence, it's just like kind of showing the sequence from the 3 different clones from the lentiviral CRISPR-Cas9 MIR21 gRNA1 transduced cell, which is like a lot to say for that, but it's just kind of like showing kind of like what those sequences look like by using CRISPR. Is like it's clearly showing a mutation, so it's clearly doing something. And then like it's showing like it's showing the actual deletions, it's showing the actual mutations up here, and like what the deletions look like, or what, you know, adding looks like. So there's different things like that. Like it's very obvious that it can be used to do it, or to like create those modifications.